Hello everyone, welcome to this tutorial on NGS basics and quality control in bioinformatics using Galaxy. Before starting with, let me tell you about the Galaxy server. Galaxy is an open source GUI based no coding required platform where you can perform various kinds of bioinformatics work, and this video will be useful for the beginners who are working with DNA or the RNA sequencing data. And in this tutorial, we'll be performing some basics in the NGS data analysis without coding and only a point-on-click analysis. Now, let us talk about what is NGS and why quality control matters. NGS data analysis is the step-by-step process of checking, cleaning, aligning, and interpreting the sequencing reads to find the genetic variants, gene expression patterns, or other genomic informations. And this usually involves several methods, like it starts with the quality control where we check the raw reads for errors, and then comes the trimming where we remove the poor quality parts or adapter sequences, and next is the alignment where we match the reads towards the reference genome, and variant calling or quantification. Here we detect the mutations in a gene, or we can measure the expression levels. Next is the annotation and interpretation. So we annotate the reads. After annotating, we find the results and we check that results whether it is any associated with genes, traits, or the diseases. And before using this data for analysis, we must assess and improve its quality. So this is done by using tools like FastQC and Chemomatic. FastQC is a tool that helps to inspect the quality of your reads, and it also helps to identify the low quality reads, adapters, and GC bias. All these things can be identified by using this tool, FastQC. And next is the Chemomatic Adapt. So this is a tool that trims or removes the low quality reads and adapters to process into a good quality read so that you can use it for the downstream process like alignment, variant calling, gene expression, and the functional analysis. With the right tools, we can transform the raw reads into an actionable insights. Now let me show you the demo. So to begin with, I want to tell you how to fetch the RNA or the DNA sequencing data, which is directly from the NCBI, by using the inbuilt tool in the Galaxy. So here you can type faster. Yeah, so this is the tool which is faster download and extract reads in the FASTQ format from NCBI SRA. So this tool will directly fetch the RNA or the DNA sequencing data which is directly from NCBI in the format of FASTQ. So I'm just going to click this tool, and it asks several questions like select input type. So let it be SRR accession because we're going to enter our SRR accession number. So SRR 17676981. So this is a SRR accession ID which we're going to work with. So I just going to give run tool. You need not make any other changes. Let it be default unless until you need any formatting. So I'm just going to click this run tool over here. Yeah, so as soon as I give this, it says that it produces 4 outputs. One is 4 files like FASTQ dump log file, parent data, single end data, other data. So it will be processed under this history section, and once the job is completed, all these folders will automatically change to green. And one more thing I want to mention here is, if you have already downloaded any sequencing data in your system, you can directly fetch those data and import it in this Galaxy by using this upload option. So guys, now we got all the files and our data is a parent data. So here you can see a list with one fastqsanger.gz pair, which means our parent data has been downloaded and it is compressed in a fastqsanger.gz pair, and rest of the things it shows a list with 0 data set in the case of single end data, and other data also shows it is 0 data sets, which tells that there is no results here. Since our data is a parent data, we got the data which has been downloaded in this part. And fourth file is the FASTQ dump log. So this is a log file that just tells about what commands were run behind the scenes, and if any error or warnings occurred, it will be projected, and it is useful for debugging or verifying if the job is finished correctly or not. So all these can be checked by this log file. Now we're going to work only with the parent data. So I'm just going to click this, and here you can see that the SRR accession ID with the data has been downloaded here, a pair with 2 data sets. If I click again here, you could see 2 files, which is forward and the reverse. Yeah, now you can download it in your system by clicking this option. The entire data will be downloaded. So guys, this is how we need to fetch our FASTQ download and extract reads in the FASTQ format from NCBI SRA tool. So I hope you can do it by yourself. Now, let me begin with one more thing called quality check. So for that, let me type a tool called FastQC read quality reports. So this is a tool that we use for checking the quality of our reads. So since we got two files over here, let us work with the forward reads now. So I'm just going to click this option. So the file has been uploaded here successfully, and you need not make further changes here for the quality check. And now just click run tool, and the file will be processed here in this history section. So we get two folders which has been running here. One is raw data and the other one is web page. So now the job is completed. Now you can see the raw data's details over here by clicking this display option. And one more thing is we get web page. So if you click this display option, it will project all the FastQC report. Here you can see several modules has been failed here, including pair-based quality and GC content and overrepresented sequences. All those modules has been failed, so which you can get the report and visualize by clicking this folder. And if you scroll down even more, you can get all the graphs according to the module. So for example, per base n content graph is here, and sequence length distribution, sequence duplication levels, overrepresented sequence. So all the details about this modules has been visualized here. So you can take this as a result. And if you come to per base sequence quality, here you can see that there are several reads which are entering into this pink and the pale pink color. So this tells that our reads to be go for trimming, and it needs to be cleaned properly. So for that we will be using a tool called Trimomatic. So this is how we have to fetch our data and do for quality check to check the quality of our raw data. Now let me show you how to use the Trimomatic tool in order to clean up the raw data. So for that I'm just going to type Trimomatic. This is the Trimomatic flexible retrimming tool for Illumina NGS data. I'm just going to click this. So this asks several options like single end or parent reads. So ours is a parent read, so that I'm giving two separate input files here, this option, and it asks for two inputs. First one, we can upload our forward read, and for the second one we can upload reverse. Yeah, now both the files have been uploaded successfully. And for the perform initial Illumina clip steps, so for that we need to give yes option. So this uses a tool called cut adapter to trim our poor quality bases. And now as you scroll down, it will ask for adapter sequences to use. So for this we need to click the third option for the parent data, and let the rest of the thing will be default. Yeah, as you scroll down, it asks like Trimomatic operations, and here you can give the sliding window as default. And now just click this plus option in order to add up some more Trimomatic operations in order to have the better improvements with our raw reads. So as you scroll down, you will get this option like minlen. So this minlen will do drop reads below a specified length. So here you can give 50. And again click this plus option and add one more Trimomatic operations, and here you can give leading. Okay, let it be 3. And next option, here you can give trailing. So each Trimomatic operations has its own function, so accordingly you can customize. So let the rest of the options be default, and as you scroll down you can get the entire detail about all the Trimomatic operations and its function. So you can modify it according to your need. So now let me keep only this for the changes. You need not make any further changes. Now you can just give run tool. Yeah, it says that we get 4 outputs now. One is R1 paired, R2 paired, R1 unpaired, R2 unpaired. Let us go only with the R1 paired and R2 paired, which is our forward and the reverse reads. So the files are being processed. Yeah, so the job is done. Now let me check with the 9th folder, which is trimmed data of forward reads. Here I'm going to click this display option. This will display our trimmed result. Yeah, we got our result here. So now after we do Trimomatic or after we do trimming, we need to recheck. We need to recheck with the FastQ quality control tool in order to check for the improvements. So for that I'm going to check it again by giving a tool called FastQC read quality reports. I'm just going to fetch this trimmed sequences over here in order to check for its improvements. So here I'm going to click this option and give like Trimomatic on SRR 1766791 forward. Yeah, this one. So I have uploaded the file, which is a trimmed file of the forward reads. Likewise, you have to do the quality check for the reverse reads also. For now, I'm doing only with the forward. So I'm just going to click Run tool. The files are being processed. And now we can check for the improvements after we perform trimming with our trimmed raw reads. So now we got the result with 2 files. This is again the raw data and this is the web page. Let us see for the improvements. So guys, as you scroll you can see there are reads which are touching only in this green part. So this shows it has trimmed properly and we get a good quality sequence reads here. So guys, this is how we need to perform Trimomatic. See this entirely. Now the reads are good. Now the reads are with good quality reads. So now you can directly use it for the downstream process like HiSat2 feature counts and gene expression. So guys, this is how we need to do Trimomatic by using the Galaxy and it is just simple as that. To conclude, by using this Trimomatic and FastQC in the Galaxy, we improve the quality of our sequencing data. So this ensures that downstream analysis like alignment, variant calling or gene expression are based on high confidence reads which can be done using this Galaxy and it is just simple. I hope you find this video helpful and thanks for watching.